Hello, in this session we'll study about carbohydrates. We'll understand what carbohydrates are, we'll understand the function of carbohydrates and the different types of carbohydrates. Now what are carbohydrates? Carbohydrates are biomolecules. The first part that we need to understand is that they are biomolecules. They're made up of carbon, hydrogen and oxygen, and they are hydrates of carbon, which means that each carbon atom has a molecule of water associated with it. Let us look at the molecule glucose. It has 7 carbon atoms. So if I draw, 6 carbon atoms, 3, 4, 5, 6. If I draw 6 carbon atoms, each of them has a water molecule associated with it. So this is how it goes. And if you now look at what I've drawn, is I've drawn one carbon atom and each of them is associated with the water molecule. If I add up the number of atoms of a given element, then I'll find that I've got 6 carbon atoms. 1, 2, 3, 4, 5, 6. I've got 12 hydrogen atoms. 2, 4, 6, 8, 10, 12. And then I've got 6 oxygen atoms. 1, 2, 3, 4, 5, 6. So similarly, you can solve the example of heptose, which is a 7 carbon atom monosaccharide or carbohydrate, a simple carbohydrate. So much for the definition of a carbohydrate. Now, what is the importance of carbohydrates? Carbohydrates are important sources of energy. You can look at the example down here. This is a molecule of glucose, and glucose is used by most of the forms of life as a basic source of energy. The oxidation of glucose, the addition of oxygen to it, will release a good amount of energy, which is then stored in ATP molecules. Another important aspect or function of carbohydrates is that carbohydrate molecules act as storage molecules for energy. Let us have a look at an example. Starch, for example, here. Starch is a primary energy storing molecule present in plants. In case of animals, glycogen serves the same purpose, glycogen. Now anyways, this is the diagram. This is an electron micrograph of chloroplast, inside of which you can see these are the thylakoids stacked on top of each other. This is the place where photosynthesis takes place, and this is the place where carbon dioxide and water are combined to make glucose. And this glucose is then a lot of these glucose monomeric units are then polymerized to form starch, and these are the starch granules here. The third function of carbohydrates is that carbohydrates in plants, in fungi, in bacteria, and even in animals provide structural support to the organism. In case of plants, in the cell wall, a carbohydrate called cellulose is present. Also, another carbohydrate called hemicellulose is also present. Again, another carbohydrate molecule called lignin is also present, and all of these carbohydrate molecules, in case of plant cell walls, will impart integrity. You can see in this particular diagram here, all these fibrous structures that you see, all of these fibrous structures that you see here are cellulose fibers. In case of fungi, the cell wall of the fungi is made up of a certain carbohydrate called chitin, and the same carbohydrate called chitin also makes up the exoskeleton of a couple of animals, such as the beetles, which is an insect. Again, many other insects, like you got flies. The exoskeleton of a lot of these animals is comprises of chitin. The shells of the crabs and the prawns are also rich in the carbohydrate called chitin. In case of bacteria, the cell wall contains a special carbohydrate protein complex, which is known as peptidoglycan. Let's write it down here, peptidoglycan. So this is basically a combination of particular carbohydrates and amino acids. So what we can see is carbohydrates are also an important part of the structural components of various forms of life. One more important function of carbohydrate is that they are components of the cell membrane. and also many other important biomolecules. Now in the cell membrane we find carbohydrates in the form of glycoproteins and glycolipids. So glycoproteins and also in the form of glycolipids. And in the cell membranes, what these two molecules do, they act as receivers and transmitters of cellular communication between two cells which are close by or distant from each other. Also, these particular molecules act as special molecules which are responsible in cell-to-cell communication and identification of self from non-self, etc., etc. Carbohydrates are also an important component of the two very important molecules called DNA and RNA. Now DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. Now nucleic acids are the molecules that carry information from one generation to another and also express this particular information within an organism. So DNA, the deoxyribonucleic acid, it is rich in a particular kind of a sugar which is known as deoxyribose, and this pentagonal structure that you see here, this blue structure, is deoxyribose sugar, which is a component of DNA. RNA, which is a genetic material in a couple of virus but not in other organisms, but plays a very important role in the formation of or synthesis of proteins, is RNA, and RNA contains ribose sugar. So much for the functions of carbohydrates. Now let us try to understand how do we classify carbohydrates. Now carbohydrates are classified on the basis of the number of fundamental units they are made up of. For example, the fundamental unit that makes up a protein is known as amino acid. So amino acids, they make up proteins. They are the building blocks of proteins. Cell is a building block of a living organism, a multicellular organism or even a unicellular organism, that is a building block. Similarly, carbohydrates can be classified into three types: monosaccharides, let's choose a different color, into monosaccharides, oligosaccharides and polysaccharides. And what are the bases? How many fundamental units are present? Now the fundamental unit or the building unit of carbohydrate is the monosaccharide itself, and let us first of all see a monosaccharide. Now here you got three monosaccharides: ribose, glucose and fructose. Now there are many other monosaccharides, a large number of them are present, but we are taking an example of these three. Now you can see that a monosaccharide is the simplest kind of a carbohydrate. And what kind of a carbohydrate is a monosaccharide? What is the other important property? A monosaccharide is made up of a minimum of 3 carbon atoms to 8 carbon atoms. Let us have a look at this. So ribose is made up of 5 carbon atoms, if you see. So we got carbon atom number 1, 2, 3, 4 and 5. Similarly, glucose is made up of 6 carbon atoms, 1, 2, 3, 4, 5, 6. And likewise is the case with fructose, which is made up of 6 carbon atoms. So any carbohydrate that contains a minimum of 3 or a maximum of 8 carbon atoms is known as a monosaccharide. Now these monosaccharides can combine in different ways. A monosaccharide can combine with another monosaccharide. For example, glucose can combine with fructose, or fructose can combine with another fructose. And the combination of these monosaccharides will give rise to another group of carbohydrates called the oligosaccharides. So the first group of carbohydrates is a monosaccharide, which are nothing but the simplest of the carbohydrates, made up of 3 to 8 carbon atoms. And now going on to oligosaccharides. Oligosaccharides are carbohydrates which are made up of 2 to 10 monosaccharide units. I have type 20 here, but this is 10. 2 to 10 monosaccharide units, a minimum of 2 or 3 or 4, 5, 6, 7, 8 up till 10 monosaccharide units can combine together to form a larger sized carbohydrate, which is called an oligosaccharide. Polysaccharide contain more than 20 monosaccharide units bonded to each other. Right. Now let us first of all have a look at a couple of monosaccharides. So before we get into monosaccharides, let us understand how do we write the structures of monosaccharides. Now let us write the molecular formula for glucose. So So this is the molecular formula for glucose. I hope everybody knows it, C6H12O6. Now this particular glucose molecule, how does it actually exist in nature? So this is the way, this is the form in which it actually exists, and this is the cyclic form. This is called the structural formula. We are going to write down the structural formula. There are two ways of writing down the structural formula. The first way of writing down the molecular formula is called a Fissure projection, and it is called so because it was devised by Emil Fischer. Now here we see this is the Fissure projection. So this chain, the long chain structure of glucose is called the Fissure projection, and this describes the 6 carbons. So this is the first carbon, the second carbon. So C is not written here. It is understood that we have a carbon atom here, and then this is carbon atom number 3, carbon atom number 4, number 5, and the 6th carbon. The terminal carbons are always depicted as C's with the letters, right? So this is the open chain structure or structural formula for glucose according to the Fissure projection. In reality, however, if you look carefully, carbon position number 1 is an aldehyde. In this case, it's an aldehyde. It's a functional group. I hope you've studied this in organic chemistry. Now the aldehyde present at position 1, carbon position number 1, has the ability to interact with hydroxyl functional group present at carbon position number 5. Now this aldehyde can also interact with carbon position number 4 in case of other monosaccharide. They can also interact with many other hydroxide groups within the chain. It depends upon the length of the monosaccharide. Anyways, so what happens in actual situations, in real life situation, or in aqueous solutions, to be very specific, the OH group of the aldehyde, it interacts with OH group at position number 5 or 4, and what happens is that a molecule, when they interact together, a molecule of water is liberated. So this means this D-glucose or the glucose molecule dehydrates and a bond is formed. So now we have this hydrogen is gone from here and this OH is gone from here, right? And they have joined together to form a water molecule. What do we have left inside? We are left out with an oxygen atom. So this carbon is bond. This carbon here is bonded to this oxygen via a single bond, and now the valency of oxygen, because of its valency, it is going to combine to this. It is going to bond with this particular carbon, and a bond is established between the oxygen connected to carbon on the 5th position and carbon number 1, and a bond is formed like this, and this molecule acquires a circular or a cyclic shape, and this is that cyclic shape. And let us decipher, let us equate these two structural formula. And so this is carbon number 1, this is carbon number 1, and on carbon number 1 we have an OH. Okay, so we have an OH. Let's say this is that OH, and carbon position number 1, we got an OH, and then we have this oxygen bonded here, and then this is connected to carbon number 5, which is out here, and to carbon number 5 we got the 6th CH2OH group out here, right? So it is the interaction between the aldehyde group here. Let's reduce the size of the eraser. So the interaction between the aldehyde group on carbon position number 1 and the hydroxyl groups at carbon position number 5 or 4 turns the glucose linear molecules into a cyclic form. Now the two structural formulas are known by specific names. So this linear structural formula is called a Fissure projection, and the cyclic structural formula is called a Haworth projection. Anyways, moving forward, let us come back to monosaccharides. Now monosaccharides, these are the different classes. So monosaccharides can further be grouped or classified into on the basis of two types. Monosaccharides can be grouped on the basis of the functional groups present on them, and monosaccharides can also be classified into different groups based on the number of carbon atoms present in them. Let us look into the details of it. Now let us classify monosaccharides on the basis of functional groups present in them. So here we've got two different monosaccharides. You see, we only have 3 carbon atoms, a minimum of 3 carbon atoms. How, and this carbohydrate also contains 3 carbon atoms. But how do these two carbohydrates differ from each other? If you see on this particular end, we have an aldehyde group. Now aldehyde groups CHO, CH double bond O, this is an aldehyde functional group. It is always present onto the terminal ends on the sides. So the monosaccharides that contain an aldehyde functional group are classified or categorized as aldoses. If you look at this particular monosaccharide, this does not contain an aldehyde group. It does not have a CHO group. There is an OH group, but there is no CHO functional group. Instead, this particular carbohydrate has a C double bond O functional group. And this carbon is attached to two carbons on the other ends, so as to satisfy its tetravalency. Anyway, so this particular functional group COO is C double bond O, is known as a ketone. And the monosaccharide that contain a ketone functional group are known as ketoses. Now let us have a look at a couple of examples of aldoses and ketoses. Let's spot them. So here we've got a large number of aldoses. And why do I categorize these carbohydrates as aldoses? Because see, this is the functional group CHO, aldehyde functional group. See, aldehyde functional group, CHO, aldehyde functional group. Pick out anyone, look at any one of them and you'll find aldehyde functional group. So all of these monosaccharides, and why are they monosaccharides? Because they contain 3 to a maximum of 8 carbon atoms. Does this contain 8? 1, 2, 3, 4, 5, 6. So in this plate we have only carbohydrates ranging from 3 carbon atoms, or monosaccharides ranging from 3 carbon atoms to 6 carbon atoms. So we have 3 to 6 carbon atom chain long monosaccharide carbohydrates present in this particular plate. Anyways, so these are examples of aldoses. Now let us look at a couple of examples of, and let us also name a couple of aldoses. So the simplest of the aldose is called a glyceraldehyde. It's a 3 carbon aldose. Glyceraldehyde. Simpler one will be ribose. So you got ribose, you got glucose, and you can remember one more, mannose. This is easier to remember, the nose of a man, mannose. So you got glucose, mannose, and ribose are examples of aldoses. Let us look at a couple of examples of ketose monosaccharides. So dihydroxyacetone, see here, we have a ketone group, C double bond O. Very important to remember, C double bond O, ketones. These are all ketoses. These monosaccharides are all ketoses, which means that they contain ketone functional group. And well, 1, 2, 3, 4, 5, 6, again 3 to 6 carbon atom long monosaccharides have been depicted in this particular plate. And also let's figure out a couple of examples of these ketoses. So here is an easier one. Fructose is a ketose, fructose. And then xylulose, you can remember one more as xylulose. Right, so these are examples of, or ribulose. You can remember 1 or 2 examples of ketosis. Coming back to monosaccharides, once again the classification of monosaccharides. So what have we done so far is we have seen the different types of carbohydrates, how do we classify them, and also now we are looking into monosaccharides and how do we further classify monosaccharides. What are monosaccharides? How do we classify them? Now on the basis of functional groups, you classified them as aldoses and ketoses. You must remember at least one example, glucose and fructose is easy. Now monosaccharides can also be classified on the basis of number of carbon atoms present in them. Now here we are. Monosaccharides can be classified on the basis of numbers of carbon atoms in them. If a monosaccharide has only 3 carbon atoms in it, we call it a triose. If it has 4 carbon atoms, it is called a tetrose. If it is 5, we call it pentose. 6 is hexose, 7 is heptose, and 8 is an octose. And let us look at a couple of examples here. Now, these are all aldoses because they contain an aldehyde group, but what, on the basis of number of carbon atoms, what kind of a monosaccharide is this? And so this contains 1, 2, and 3 carbons. 1, 2, and 3 carbon atoms here. So this is a triose. This is a triose. Let us look at this one. 1, 2, 3, and 4 carbon atoms. So what should we name it? How should we classify it on the basis of number of carbon atoms? Tetrose. Tetrose. Let us have a look at this. 1, 2, 3, 4, and 5. So this is a pentose. This is a pentose monosaccharide. Right. Similarly, also let us and this one has a 6, 1, 2, 3, 4, 5, and 6 carbon atoms. So this is supposed to be a hexose. This is a hexose monosaccharide. Again, in the case of ketosis, also it's the same. So we've got 3 carbon atoms here. What kind of a monosaccharide is this on the basis of number of carbon atoms? This is a triose, and this has 1, 2, 3, 4. This is a tetrose. And 1, 2, 3, 4, 5. This is a pentose and hexose, and you can similarly have heptose and octoses. Right. So this is how we classify monosaccharides. Let's go back once again and try to recapitulate. So monosaccharides can be classified on the basis of functional groups into aldoses or ketoses. And monosaccharides can also be classified into triose, tetrose, pentose, hexose, heptose, and octose on the basis of how many carbon atoms do the monosaccharides contain. Now moving forward to oligosaccharides. What are oligosaccharides? Oligosaccharides are those carbohydrates which contain from 2 to 10 monosaccharide units. 2 to 10. Now an oligosaccharide which contains only two monosaccharide unit is called a disaccharide. A couple of examples are maltose, lactose, and sucrose, and let's have a look at that particular slide which contains more details. So here are 3 disaccharides: sucrose, lactose, maltose. Why are they disaccharides? Because sucrose is made up of 2 monosaccharides, glucose and fructose. So this is glucose, this is fructose. Lactose is made up of galactose and glucose. This is galactose and this is glucose monosaccharide. And maltose is made up of glucose and another glucose, identical 2 glucose units. So maltose, this is also glucose, this is also glucose. So disaccharide is the simplest of the oligosaccharide. Oligosaccharides are carbohydrates which contain 2 to 10 monosaccharide units. Now one important thing that we must learn from these particular diagrams is that the 2 monosaccharide partners are connected to each other via a bond, and you see this bond in the center. This particular bond which connects 2 monosaccharide units is called a glycosidic bond or a glycosidic linkage. So glycosidic bond or a linkage. So let us try to understand how are glycosidic bonds formed. Now the reaction that gives rise to a glycosidic bond is known as a condensation reaction. And so here what do we have? We have here a molecule of beta D-glucose, and again we have alpha D-glucose, and we have another molecule of alpha D-glucose. Now at the moment, let us not try to make it more complex. Why are they called alpha glucose? Why is it a D-glucose? Is something that you will learn in your organic chemistry classes. But for the moment, you need to just understand that how a glycosidic bond is formed. We'll certainly talk about alpha and beta glucose. What do we mean by that? What are alpha 1,4,1,6 linkages, etc., because that will be helpful to us. So let us look at this particular molecule. So this is carbon position 1. I hope you can see C1. This is the first carbon. This is the second carbon. This is the third carbon. This is the fourth carbon. This is the fifth carbon, and this is the sixth carbon of the first molecule. The second glucose molecule. In the second glucose molecule, this is the first carbon, this is the second carbon, this is the third carbon. Here they have already typed out fourth. This is the fifth carbon, and this is the sixth carbon. Now, for the bond to be formed between these 2 glucose molecules, the hydroxyl group, now remember the position of hydroxyl group. The hydroxyl group attached to carbon position number 1 is to the bottom, not to the top. In some cases, OH can be on the top, and at the bottom you can have an H. But in this particular case here, what do we have is you've got an OH group down and 1 H is to the top. It has been written to the side, but to make it more clear, we can write it here, right? It has been written here, but you can write it to the top. So when the hydroxyl group attached to carbon position number 1 is oriented to the lower side, we call that particular monosaccharide as an alpha monosaccharide. In this case, the monosaccharide is glucose, so we call this as an alpha glucose. If this was to be beta glucose, then this OH would be to the top, and at the bottom you will have the hydrogen atom attached to the carbon position 1. The most important thing is we only define alpha and beta monosaccharides on the basis of position of hydrogen and hydroxyl group with reference to carbon 1, the first carbon, not with respect to 4th, 3rd, 2nd, 5th or 6th. Right. Anyways, so I hope you've understood what do we understand by alpha and beta monosaccharides. So why is this alpha glucose? Because in this particular case, the OH group is to the bottom and hydrogen is to the top. For simplicity, I'm writing it to the top. I'm bonding it to the top. Now, when the bond formation takes place, the bond formation takes place between carbon 1 of one of the glucose units and carbon 4 of the other glucose units. Now, the two OH groups come closer together, and in the presence of a certain enzyme, both of them undergo something called as a condensation reaction, and during this reaction a molecule of water is liberated out. So this hydrogen is removed, this O is removed, and one more hydrogen is removed, and what are we left out with? We are left out with one oxygen atom, and this oxygen atom, then let's use a marker. So this oxygen atom is already bonded to this carbon 1, and this oxygen atom also gets bonded to carbon 4. And this particular bond here, let me just highlight this bond here. Let's use a certain different marker. So this bond that is formed here, this one here, between carbon 1, carbon 4 via the oxygen atom here, this is called a glycosidic bond. Okay, and let us have a look here. Where do we see? Let us have a look here. This is exactly what a glycosidic bond is. And I told you, when these two hydroxyl functional groups come close by, they undergo a reaction called condensation reaction. Why condensation reaction? Because the two molecules are condensed together. They are brought together, close together to each other. And also this is a dehydration reaction because a molecule of water is removed, dehydrated, right? And what is left behind? An oxygen atom, which then bonds or links the carbon 1 and carbon 4 of the other. And this bond that connects two monosaccharide units together is called a glycosidic bond. This is important for all of us to remember, for you to remember particularly. So a glycosidic bond, let's highlight this here. So this is the glycosidic bond, and here is the glycosidic bond, right? Now, I hope you've understood a couple of concepts from here. First of all, I hope you will be able to differentiate between alpha and beta monosaccharides, and this is very important. Saccharides, and also I hope you can now figure out what a glycosidic bond is. And a glycosidic bond is basically a result of condensation reaction in which dehydration occurs, and this is an enzymatic process, enzyme driven process. Alright, so maltose is also one of the components that we ingest as a part of our nutrients that we intake. Now maltose is also broken down by our body with the help of an enzyme called maltase. Now what does maltase do? The objective of maltase is to release monosaccharide units, is to release these glucose units which can be used by our body. So in order to get these glucose molecules back, this glycosidic bond must be broken. So the enzyme maltase, what it does is, it, in the presence of water molecule, this is called a hydrolysis reaction, because we are adding water to lyse, to break this bond. So this is a hydrolysis reaction. So maltase hydrolyzes maltose. Maltase hydrolyzes maltose and breaks this glycosidic bond and reverses the condition back to this state. Right? I hope our discussion about glycosidic bond, the condensation reaction, formation of glycosidic bond, and the breaking of glycosidic bond is amply clear to you. Let's proceed further. Now this is a little bit technical, but this is just for a little, this is here only to introduce you to the fact that there are wide varieties of glycosidic bonds. Please always remember that the glycosidic bonds are always formed between carbon 1 and carbon 4 and OHs, the hydroxyl groups. The glycosidic bonds involve hydroxyl groups of the two monosaccharide units. This is what you must remember. Now in some cases we have an alpha monosaccharide involved. In some cases we have got a beta monosaccharide involved. In some cases the bond is formed in this particular fashion where the alpha monosaccharide, the hydroxyl of alpha monosaccharide combines with the hydroxyl of the adjacent monosaccharide unit to form a glycosidic bond. In that case we call it an alpha linkage. And if it, why do we call it 1,4, alpha 1,4? Because carbon 1 and carbon 4 are involved. If the bonding takes place between carbon 1 and carbon 6 and the alpha monosaccharide is involved at carbon position 1, then we call it an alpha 1,6 bonding, right? So there is nothing much to remember. All you need to know, the objective of telling you about this particular aspect here is that you must remember that glycosidic bonds are of wide varieties. You have glycosidic bond known as alpha 1,4, you can have alpha 1,6, you can have beta 1,4, you can have beta 1,6 glycosidic bonds, etc., etc. So you can have wide varieties of glycosidic bonds. This is all you need to remember at the moment. Alright, now what are the functions of oligosaccharides? Oligosaccharides perform a wide variety of functions, and here again what do we have is we have another structural formula or a Haworth projection of raffinose, and you can see it is made up of 3 monosaccharides: galactose, glucose and fructose. And these are this is the glycosidic bond between galactose and glucose, this is the glycosidic bond between glucose and fructose. So this is an oligosaccharide. But what are the important functions? The oligosaccharides are important in cellular signaling, which means communication between cells, cell recognition, whether what kind of a cell is this the cell from my body or is it a foreign cell, cell binding when 2 cells have to be connected to each other, transport of carbohydrates in plants. Oligosaccharides are also involved in transport of carbohydrates. They act as transport carbohydrates in plants. Finally, moving on to polysaccharides, and let us go back a couple of slides to see what polysaccharides are. So polysaccharides here are monosaccharides which contain 10, not 20, but 10 or more than 10 monosaccharides. So 10 or more than 10 monosaccharide units, greater than 10 or equal, it's just greater than 10, not 10, greater than 10 monosaccharide units, when they are bonded together via glycosidic bond, we get a polysaccharide. Now polysaccharides are of different types: homopolysaccharides, heteropolysaccharides. Homopolysaccharides have similar monomeric units. For example, if they are made up of glucose, there is just glucose monomers connected to each other. Heteropolysaccharides will have different types of monomeric units being used as building blocks. Anyways, let's proceed further. Let's look into the details of polysaccharides. And so let us look at the 3 major examples. Cellulose is an example of a polysaccharide, starch is an example of a polysaccharide, and glycogen is an example of a polysaccharide. So cellulose we see in plant cell walls, starch is a storage carbohydrate in plants, glycogen is also found in plants. We'll look into the details of each of them in a little while. Now, what is cellulose made up of? What kind of monosaccharide? Now remember, cellulose is a homopolysaccharide. It is made up of beta glucose. Let us have a look at beta glucose. So here we've got cellulose, and these monomeric units are beta glucose units. Let us have a look at this particular unit here, or so let's take a moment here. Yeah, so this is a glucose. Here we have glucose. So this is carbon 1, carbon 2, carbon 3, 4, this is 5th carbon, and here we got the 6th carbon, right? So we got the 6 carbons here. Now look at carbon position 1, the first carbon. Look at the first carbon. The hydrogen is at the bottom, and on the top we have a bonding from the top, which means this is the place where we had the OH group, the hydroxyl group. So what did we learn about beta monosaccharides? So beta monosaccharides have their hydroxyl groups oriented towards the top, and that's why this kind of a glucose is called a beta glucose. So cellulose, the building blocks of cellulose, or the subunits, the building block is beta glucose. The first point that you must remember, cellulose is made up of beta glucose. This is also beta glucose, right? And here again, but This is an inverted molecule, so you don't have to be bothered much at this level. Only remember that beta glucose units are linked to each other via glycosidic bonds. This molecule has been flipped to the other side, and this is so. Anyways, looking at the next part, what is the nature of bond? This is beta 1,4 between carbon 1 and this is carbon 4. The carbon 1, carbon, this is wrongfully typed as O, this should be C. So carbon 1, carbon 2, carbon 3, and this is carbon 4. So this is beta 1,4 linkage. Beta, because this is beta glucose, beta 1,4 linkage. So the bond is 1,4. And what else? So is it a branched molecule? So here, let us have a look at the diagram here. This is a straight chain. Cellulose is a straight chain polysaccharide. Even you can look here also. So this is the cell wall. In this particular diagram, we have a leaf. Inside the leaf, we have got cells. The cell, we have been shown the cell walls. The cell wall has a large number of cellulose microfibrils. If you take one of the microfibrils, it will contain large number of cellulose molecules. And these are cellulose linear chains. There is no branching, right? There is no branching between cellulose molecules. Now, some important aspects about it. The cellulose is an organic compound made up of beta glucose units. So we've done this. The first point that you must remember about cellulose. The beta glucose unit differ from alpha units, and we've done this in the orientation of their hydroxyl group. Now, cellulose cannot be digested by human beings or most of the animals. Why is it so? Because our body, our digestive system does not contain any enzyme which can break the beta 1,4 glycosidic linkage. This is the beta 1,4 glycosidic linkage. This here is beta 1,4 glycosidic linkage, and we do not have the enzyme to break this down. However, this particular linkage can be broken down in the intestines of ruminants. Now, ruminants are the herbivores which have a part of the stomach called rumen present in them, and they have special microbes present out there in that part of the stomach, microbes. And these microorganisms produce enzymes that can break this linkage, and that is the reason why ruminants can digest cellulose. Cellulose can be digested by them. Unfortunately, we cannot. Anyways, what are the different commercial usages of cellulose? Cellulose is used to make paper, right? And cellulose is extensively used in textiles. So all the cotton fabrics and the jute bags, etc., that you use are rich in cellulose. Cellulose is also used in pharmaceutical industry to make the bulk in which drugs are embedded. Also, it is used to make explosives, and it is also used in toothpaste. You can check out the labels of your toothpaste and you will find something called as carboxymethyl cellulose in it. And these are the different usages of cellulose. Moving to the next polysaccharide, we have glycogen. Now, glycogen is compared to, okay, let's go back to our comparative table of comparison, and we are coming here. So this is what we have here is glycogen. Now, glycogen is found in animals only because it is a storage polysaccharide present in animals, right? And again, what are the building blocks of glycogen? Let us have a look here. So all of these are alpha glucose units. Let us have an alpha glucose. See, the OH is down here, and that's why the bonding is at the bottom, right? So these are alpha glucose units. So alpha glucose units are connected to each other by two different kinds of bonds. So let me just rub it off and let us now focus towards the bonding. So in glycogen, we find two kinds of bonds. First, we find this bond here. This is a glycosidic bond. It is between carbon 1 and carbon 4 of the adjacent. Right. So this is called an alpha 1,4 glycosidic bond or a glycosidic linkage. So the straight chains will contain alpha 1,4 linkages. However, if you see, the straight chains also have cross linking, which means the long linear chains are also connected to each other via another glycosidic bond. This is also a glycosidic bond, but this glycosidic bond is between carbon 1, if you look carefully, and the 6th carbon of the adjacent glucose unit. And that's why this particular glycosidic bond is called an alpha 1,6 linkage. So glycogen contains two types of glycosidic bonds, alpha 1,4 linkage and alpha 1,6 linkage. The branching in glycogen is because of alpha 1,6 linkage. And what do we see here? Compared to cellulose, glycogen is a highly branched structure, extremely branched. There is not one branch, two branch, but each branch further branches into many branches. So it is a highly branched structure. Now, what we need to understand, what should we also know about glycogen is that glycogen is a polymer made up of glucose. So whenever we digest food in the intestines, we got large amount of glucose which is absorbed by the intestinal walls and is dropped into the blood vessels. From there the blood carries glucose to the various parts of the body. Now the body has the mechanism to determine the amount of glucose in the blood. If the amount of glucose rises above normal in the blood, a hormone called insulin is released by the pancreas. Now this particular hormone called insulin acts on the cells of the liver. This particular insulin hormone acts on hepatocytes, cells of the liver. Now hepatocytes, what do they do then? They take these glucose molecules inside. They start absorbing the glucose molecules on the inside, and then they start connecting these glucose molecules together. They make large number of these straight chain glucose molecules, and then they will link them. And what do they form? They form a large molecule called glycogen. So glycogen is made when there is excess of glucose in the blood, and this glycogen is then made within or inside the hepatocytes where it is stored. So this is a storage carbohydrate. When the concentration of glycogen falls, when the concentration of glucose in the blood falls down, then another hormone called glucagon is released from the pancreas, from the cells of the pancreas, and this particular hormone, it again acts on hepatocytes. Hepatocytes, the same cell. Now these hepatocytes, they have, what do they contain? They contain glycogen molecules. And glucagon, when they act on hepatocytes, the glucagon molecule is, the glycogen molecule, this particular molecule is broken down to release glucose units. Right. So much about glycogen that you should know. What kind of linkages, what kind of glycosidic bonds are present in between alpha glucose subunits in glycogen, alpha 1,4, alpha 1,6, and it is highly branched. It is a storage molecule. Where is it stored? Where is it found? In animals. Where in animals? In hepatocytes, and a couple of other things, the role of insulin and the role of glucagon. Proceeding to our last important polysaccharide, which is starch. Let me just bring down this toolbar. Now starch is the major storage polysaccharide present in plants, and this is very important for us to know. It is just a minute. So here it is. So starch is the major storage polysaccharide present in plants, as we saw in our first slide. So, and starch is made up of two components. One is called amylose and the other is amylopectin. So starch is not a simple molecule, it is a complex polysaccharide. It is made up of two parts, amylose and amylopectin. Now amylose is a straight chain polysaccharide, whereas amylopectin is made up of straight chains which are cross-linked, just like glycogen was. Right. Now amylose is made up of glucose. What kind of glucose units? Let's have a look here. So amylose is again made up of alpha glucose, and amylopectin is also made up of alpha glucose. Let us see, is it true? So alpha glucose, you see, yes, absolutely. So OH group, the hydroxyl group, was actually present down here, right? And here also it was present down here. But anyways, at carbon position 1, if OH is oriented to the lower side, then we call it an alpha glucose. So So the building blocks of amylose and amylopectin are alpha glucose, and what kind of linkage is found between them? Alpha 1,4 glycosidic bond. Now amylopectin is highly branched. Again, what do we see here? This particular bond is again alpha 1,4 glycosidic bond, and this side, this cross-linking bond, glycosidic bond is, so this is also carbon position number 1. This is alpha 1 and this is carbon 6 which is attached. So this is alpha 1,6 glycosidic bond. So the structure of amylopectin is greatly similar to that of glycogen. Anyways, so starch is broken down by the activity of the enzyme amylase, which is secreted by the salivary glands, and also by the amylase secreted by the pancreas in our body. So much about polysaccharides, and I hope you've understood the meaning of carbohydrates, the different types of carbohydrates, some couple of examples of carbohydrates, and glycosidic bonds most importantly. And I hope you can differentiate between alpha and beta monosaccharides. I hope you can identify alpha 1,4, alpha 1,6 glycosidic bonds also.